All right, welcome back to the deep dive. Today we're tackling a pretty complex but super important topic.
Yeah, we're diving into the world of immunology.
Exactly. So get ready for a deep dive into how our bodies fight off all those nasty invaders,
Bacteria, viruses,
the
parasites.
whole shebang. We'll also look at what happens when our immune system, kind of goes off the rails.
Right, those immune disorders. And then, of course, how we fight back with vaccines, immunotherapy.
Oh, and we can't forget about the antimicrobial drugs we use and the whole issue of drug resistance.
Absolutely, it's all connected. We've got a really detailed chapter as our guide for this deep dive.
So let's jump right in. First up, what happens when our immune system doesn't quite work as it should?
Right, the disorders and deficiencies. It's amazing how this system that's supposed to protect us
Yeah,
can sometimes turn against us or just not do its job properly.
exactly. So let's start with hypersensitivity. Now, the name almost sounds like a good thing, right? Like
Yeah.
super immunity,
You think so?
but it's actually the opposite.
Yeah, hypersensitivities, basically when the immune system overreacts, causing damage to our own tissues.
So it's like friendly fire.
Exactly. And there are a few different ways this can happen. First, there's immediate hypersensitivity, or type I.
Ah, so this is like the classic allergy stuff.
Precisely. Think hay fever, hives, even those severe, life-threatening allergic reactions.
Okay, so walk us through what happens in the body during an allergic reaction.
Sure. Well, it all starts with these things called IgE antibodies. They're like little alarm bells.
Okay.
So the first time you encounter an allergen, like pollen or peanuts, your body might mistakenly see it as a threat
Gotcha.
and produce these IgE antibodies specific to that allergen.
So it's like the body is learning to be allergic.
In a way, yes. Now, these IgE antibodies then attach themselves to mast cells, which are like little bags of inflammatory chemicals.
So they're armed and ready to go.
Exactly. The next time you encounter that allergen, boom, it binds to the IgE on the mast cells and triggers them to release all those inflammatory chemicals, like histamine.
And that's what causes those classic allergy symptoms.
Right. Histamine makes blood vessels dilate, tissues swell, and that's why you get the sneezing, the runny nose, the itchy eyes.
So basically all those things that make you miserable during allergy season.
Pretty much. And in more severe cases, this histamine release can cause the airways to constrict, leading to asthma.
Or even that really dangerous anaphylaxis.
Exactly. That's when your whole body overreacts, blood pressure drops, and you can even go into shock.
Okay, so how do we fight back against these allergic overreactions?
Well, that's where those antihistamines come in.
Right, they block the histamine.
Exactly. And then there are anti-inflammatory steroids, which help calm down the overall inflammation.
Okay.
And then there's desensitization therapy, where you're basically trying to retrain the immune system.
Kind of like exposure therapy, I guess.
Yeah, you expose the person to tiny amounts of the allergen over time.
So the body learns that it's not actually a threat.
Right, and gradually stops overreacting.
Fascinating. Now, what about that other type of hypersensitivity, the delayed type?
Right, type IV. This one's a bit different.
Yeah, it's not that immediate explosive reaction.
No, it's a slower, cell-mediated response. It involves these T helper 1 cells, or Th1 cells.
So T cells are involved this time.
Yes, they're like the conductors of this delayed immune response.
Okay.
When they encounter an antigen they've seen before, they release these signaling molecules called cytokines.
Which are like, what, alarms?
Yeah, they trigger widespread inflammation.
Right. But this whole process takes a bit longer.
So we're talking hours or even days.
Right. Think about contact dermatitis from poison ivy, or that rash you get from certain metals.
Okay, I've definitely experienced that.
And another classic example is the tuberculin skin test.
Right, for tuberculosis.
Yeah, a positive reaction there is a delayed response.
So it's fascinating, right? Two types of hypersensitivity.
Right.
One is that quick antibody-driven reaction, and the other is this slower T-cell mediated one.
Exactly. Okay, so we've talked about allergies. Now, what happens when the immune system turns on us?
Ah, that's autoimmunity. It's when the immune system starts attacking our own tissues and cells.
So it's like the body is fighting itself.
Exactly. It's like it mistakes our own cells for foreign invaders.
And this can cause a lot of problems, right?
Absolutely. It can lead to chronic inflammation and damage to various organs.
So what's going on in the body during an autoimmune attack?
Well, it can involve those Th1 cells we just talked about, causing a delayed type hypersensitivity against our own antigens.
Okay.
Or it can involve autoantibodies, which are antibodies that actually target our own molecules.
So it's a multi-pronged attack.
Yeah, and the specific mechanisms can vary depending on the type of autoimmune disease.
Right, because there's so many different ones, right?
Exactly. Some target specific organs, while others are systemic, affecting the whole body.
Can you give us some examples of these autoimmune diseases?
Sure. Type 1 diabetes, or T1D, is a classic example of an organ-specific disease.
Right, the immune system attacks the insulin-producing cells in the pancreas.
Exactly. Then you have Hashimoto's disease, which targets the thyroid gland.
Okay.
And then there are systemic diseases like lupus or systemic lupus erythematosus, SLE.
Right, lupus can affect so many different organs.
Yeah, it can cause problems in the skin, joints, kidneys, even the brain.
So it's really a complex and varied group of diseases. Now, I remember reading something about the hygiene hypothesis. What's that all about?
Well, it's a really interesting idea. It suggests that the rising prevalence of autoimmune diseases, particularly in developed countries,
Yeah.
might be linked to our cleaner environments.
So being too clean is bad for us.
In a way, yes. The hypothesis proposes that our reduced exposure to microbes early in life
Okay.
may actually lead to an immune system that's more prone to misidentifying self as foreign.
So it's like our immune system needs a bit of training.
Exactly. It needs to learn what's a real threat and what's not.
And that early exposure to germs might actually be beneficial.
Right. It's a bit counterintuitive, but it seems there's some truth to it.
It definitely makes you think about all those antibacterial soaps and hand sanitizers.
It's all about finding that balance.
Absolutely. Now, I also remember reading something about parasites and autoimmunity.
Yeah, that's another fascinating twist.
I mean, you'd think parasites would only make things worse.
Right. But research has shown that certain parasitic worm infections, particularly helminths,
Okay.
might actually suppress autoimmunity.
So getting worms could be a good thing?
Well, it's not quite that simple, but it seems that these parasites can modulate the immune response.
Okay.
They can dampen those Th1 cells that drive autoimmune attacks.
Okay.
And they can also promote regulatory T cells, or TREGs.
And those are the ones that keep the immune system in check.
Exactly. So it's like they help to rebalance the immune system.
Wow, that's a pretty unexpected connection. What about treatments for autoimmune diseases?
There is a variety of approaches. For organ-specific diseases like T1D, sometimes it involves replacing what's missing.
Right, like giving insulin.
Exactly. And then there are broader immunosuppressive therapies to dampen the overall immune response.
Okay.
And we're seeing the development of monoclonal antibodies that target specific components of the immune system.
So more targeted therapies.
Yes, and also strategies to rebalance those T cell populations we talked about.
And of course, genetics plays a big role too.
Absolutely. Your genetic makeup can make you more susceptible to certain autoimmune diseases.
Okay, so we've covered hypersensitivity and autoimmunity. Now, what about when the immune system just gets totally overwhelmed?
You're talking about superantigens.
Exactly. What are those?
They're these special proteins made by certain bacteria and viruses.
Okay.
And they have a unique way of really revving up the immune system.
So it's like they send it into overdrive?
Exactly.
Yeah.
Unlike regular antigens, which only activate a small number of T cells,
Okay.
superantigens can activate a massive number of T cells, way more than usual.
So it's like a huge non-specific immune response.
Right. And it's all because of how they bind to those T cell receptors, or TCRs, and the MHC molecules.
Those are the ones that present antigens to the T cells, right?
Exactly. Superantigens kind of bypass the normal specific recognition.
Okay.
They cross-link these molecules and force a huge number of T cells to activate.
And that leads to
A massive release of those inflammatory cytokines.
Oh, boy.
Yeah, it's like setting off a huge alarm, even if only a small part of the system is actually affected.
And this can lead to those really serious symptoms we hear about.
Absolutely. Think about those superantigens produced by bacteria like Staphylococcus aureus and Streptococcus pyogenes.
Okay.
They can cause toxic shock syndrome and certain types of food poisoning.
Right. Those are really dangerous.
Yeah, the massive inflammation can lead to high fever, shock, even organ failure.
So it's a stark reminder of how powerful and potentially destructive the immune system can be.
Exactly.
Now let's switch gears and talk about the opposite problem. When the immune system is too weak.
Immunodeficiency. That's when the immune response is either absent or just not strong enough to fight off infections.
So people with immunodeficiency are really vulnerable.
Absolutely. Even everyday germs can cause serious problems.
And there are different types of immunodeficiency, right?
Yeah, you can have primary immunodeficiencies, which are caused by genetic defects,
Okay.
or secondary immunodeficiencies, which are acquired later in life.
So something happens that weakens the immune system.
Exactly. It could be an infection, malnutrition, even certain medical treatments.
Okay, so let's dive into some specific examples. What about severe combined immunodeficiency or SCID?
SCID is a group of really serious genetic disorders.
It sounds pretty severe.
It is.
Mm.
Basically, people with SCID have a profound deficiency of both B and T cells.
And those are the key players in the adaptive immune system.
Exactly. So they have a very limited ability to mount specific immune responses.
So their bodies can't really learn and remember how to fight off specific threats?
Right. And that makes them incredibly vulnerable to all sorts of infections.
I imagine even a common cold could be life-threatening.
Sadly, yes. Without treatment, most infants with SCID don't survive past their first year.
That's heartbreaking. But there are treatments, right?
Yes, bone marrow transplantation can provide a new source of healthy immune cells.
Okay.
And there's also gene therapy, which aims to correct the underlying genetic defect.
So there's hope for these children.
Definitely. Now, the other major immunodeficiency we should talk about is AIDS.
Acquired immunodeficiency syndrome.
Right. And we all know it's caused by HIV.
But what's the specific way HIV leads to immune failure?
Well, HIV specifically targets and destroys those T helper cells.
The conductors of the immune response.
Exactly. And as the number of these T cells declines, the immune system becomes progressively weaker.
So it can't fight off infections as effectively.
Right. And that's what makes people with AIDS so vulnerable to opportunistic infections.
Infections that wouldn't normally cause problems in healthy people.
Exactly. And it's these opportunistic infections that are often the ultimate cause of death in AIDS patients.
So it's not the HIV itself, but the weakened immune system that's the real danger.
Exactly. It's a stark reminder of how crucial a functioning immune system is for our survival.
Absolutely. Okay, so we've covered a lot of ground with immune disorders.
Right.
Now let's move on to how we can actively protect ourselves, vaccines, and immunotherapy.
Okay.
Let's start with vaccines, something I think most of us are familiar with.
Yeah.
What's the basic idea behind vaccination?
The core concept is to deliberately expose the immune system to a weakened or inactive form of a pathogen.
So it's like a training exercise for the immune system.
Exactly. It allows the body to develop immunological memory.
Okay.
So if you encounter the real pathogen later on, your immune system is primed and ready to fight it off.
That's pretty clever. And there are different ways to create these vaccines, right?
Absolutely. We have killed vaccines
Right.
where the pathogen is completely inactivated.
Okay.
Toxoid vaccines, which use inactivated toxins produced by bacteria.
Okay.
And attenuated vaccines, which use live but weakened versions of the pathogen.
So it's like a range of strategies depending on the pathogen.
Exactly. Each type has its own advantages and limitations.
Can you give us some examples?
Sure. The Salk polio vaccine is an inactivated vaccine.
Okay.
The MMR vaccine for measles, mumps, and rubella is an attenuated vaccine.
Gotcha.
And the tetanus and diphtheria vaccines are toxoid vaccines.
So quite a variety. But vaccine technology has really advanced in recent years, hasn't it?
Oh, absolutely. We now have subunit vaccines, conjugate vaccines, recombinant vaccines, even nucleic acid vaccines.
It's amazing.
Yeah.
Can you walk us through these newer types?
Sure. Subunit vaccines like the acellular pertussis vaccine use only specific pieces of the pathogen.
The parts that trigger the immune response.
Exactly. So it's a more targeted approach.
Okay.
Then there are conjugate vaccines like the Hib and pneumococcal vaccines.
Yeah.
They link a weak antigen to a stronger one to boost the immune response, especially in young children.
Interesting.
Recombinant vaccines use genetic engineering to produce large quantities of a specific antigen, like the hepatitis B and HPV vaccines.
Gotcha. And what about those nucleic acid vaccines? They're the new kids on the block, right?
They are. They use either DNA or mRNA to deliver instructions for making the antigen directly into our cells.
So our own cells become vaccine factories.
In a way, yes. And then we have plant-based vaccines, which are still under development.
So using plants to produce the antigens.
Exactly. It's a really exciting area of research.
It's incredible how many different ways we can now train our immune system. And speaking of training, why are booster shots so important?
Booster shots are crucial because they reinforce that immunological memory.
Okay.
They boost the number of antibodies and memory cells, providing longer-lasting protection.
So it's like a refresher course for the immune system.
Exactly. And we all know how impactful vaccines have been in reducing the burden of infectious diseases.
Absolutely. Now let's move on to immunotherapy, which is all about using the immune system to treat existing diseases.
Right. It's a really exciting and rapidly evolving field.
So how can we harness the immune system to fight diseases like cancer?
Well, there are several approaches. Anti-cancer vaccines, for example, can stimulate the immune system to attack cancer cells.
Okay.
And we're seeing really promising results with personalized vaccines that target neoantigens, which are unique mutations found in a patient's tumor.
Wow, so personalized cancer vaccines.
Yeah, it's really cutting-edge stuff. Then there are checkpoint inhibitors.
Checkpoint inhibitors.
They're monoclonal antibodies that target immune checkpoint proteins.
What are those?
They're basically molecules that normally prevent T cells from becoming overactive and attacking healthy tissues.
Okay.
But cancer cells can sometimes hijack these checkpoints to evade the immune system.
So they're hiding from the immune system.
Exactly. But checkpoint inhibitors block these checkpoints.
And that unleashes the T cells to attack the cancer.
Precisely. It's a really clever way to boost the body's own anti-cancer defenses.
It's like taking the brakes off the immune system. Now, I've also heard about adoptive T cell transfer.
Ah, yes. That's another exciting approach.
Hmm.
It involves taking T cells from the patient, either from their tumor or their blood,
Okay.
and then expanding them in the lab to create a large army of tumor-fighting cells.
So you're basically boosting the number of cancer-killing T cells.
Exactly. And then these supercharged T cells are infused back into the patient.
And they go and attack the tumor.
Right. And there's this really amazing form of adoptive T cell therapy called CAR T cell therapy.
CAR-T.
Yeah, that stands for chimeric antigen receptor. Basically, you take the patient's T cells and genetically engineer them to express a special receptor.
Okay.
This CAR receptor is specifically designed to recognize a particular antigen on the patient's cancer cells.
So you're giving the T cells a target to lock onto.
Exactly. And when these CAR T cells are infused back into the patient, they can very effectively target and destroy those cancer cells.
That's incredible. It's like programming the immune system to attack cancer.
It is. And it's showing remarkable results in treating certain types of blood cancers.
Now, I remember reading something about the gut microbiome and immunotherapy.
Ah, yes, that's another fascinating area of research.
I mean, the gut microbiome affecting cancer treatment. That's pretty surprising.
It is. But it seems that the composition of bacteria in our gut can actually influence how well we respond to immunotherapy.
So the bacteria in our gut are playing a role in our immune response to cancer.
Exactly. Certain types of bacteria, like Bifidobacterium and Akkermansia, seem to be associated with better responses to checkpoint inhibitors.
So a healthy gut microbiome could actually help improve cancer treatment outcomes.
That's what the research is suggesting. And there's even talk of using fecal transplants to potentially manipulate the gut microbiome and improve treatment responses.
Wow, that's really amazing. Who knew those tiny microbes could have such a big impact? Okay, so we've talked about boosting the immune system to fight disease.
Right.
Now let's switch gears and talk about those drugs that directly target those infectious microbes.
Right.
Let's start with antibacterial drugs, the antibiotics. What's the fundamental goal here?
The goal of antibacterial drugs is to kill or inhibit the growth of bacteria that are causing an infection.
So it's like a direct assault on the bacteria.
Exactly. And a key feature of a good antibiotic is selective toxicity.
Meaning?
It means that the drug should target the bacteria without harming the host cells.
Right. We don't want the cure to be worse than the disease.
Exactly. And antibiotics can be classified based on their structure, their mechanism of action,
Okay.
and their spectrum of activity.
So how many different types of bacteria they're effective against?
Exactly. Some are broad spectrum, while others are more narrow spectrum.
Okay, so let's talk about some of the different ways antibiotics attack bacteria. I remember reading about drugs that interfere with cell wall synthesis.
Right. The bacterial cell wall is a great target for antibiotics because human cells don't have one.
So it's a way to selectively target the bacteria.
Exactly. A major class of cell wall inhibitors is the beta-lactam antibiotics,
Which include...
penicillins and cephalosporins. They work by interfering with the enzymes that build the bacterial cell wall.
So the bacteria can't build their protective wall.
Right, and that makes them very vulnerable. Penicillin G is a classic example.
Okay.
But some bacteria have developed resistance to penicillin, so we now have semi-synthetic penicillins that are more effective.
So we're always having to stay one step ahead of those bacteria.
Definitely. Cephalosporins are another group of beta-lactams with a broader spectrum of activity.
Okay.
And then there are other cell wall inhibitors like vancomycin and bacitracin.
So it's a whole family of drugs targeting that bacterial wall.
Exactly.
Now, what about drugs that target protein synthesis?
Protein synthesis is another essential process for bacterial survival.
Right. They need to make proteins to function.
Exactly. And bacteria have these ribosomes, which are the machines that make proteins.
Okay.
And luckily, bacterial ribosomes are different from human ribosomes.
So we can target them selectively.
Exactly. Aminoglycosides, tetracyclines, and macrolides are all examples of antibiotics that target bacterial ribosomes.
Okay.
They bind to different parts of the ribosome and prevent it from making proteins.
So basically shutting down protein production in the bacteria.
Right.
What about drugs that target nucleic acid synthesis?
Nucleic acid synthesis is how bacteria make their DNA and RNA.
Right. Essential for survival.
Exactly.
Yeah.
Quinolones and fluoroquinolones inhibit DNA gyrase, which is an enzyme involved in DNA replication.
Okay.
And rifamycins inhibit RNA polymerase, which is involved in RNA synthesis.
So these drugs are basically stopping the bacteria from replicating their genetic material.
Exactly.
Now, what about drugs that target bacterial metabolism?
Right. Those are called antimetabolites. Sulfate drugs are a classic example.
Okay.
They block the synthesis of folic acid, which is a crucial molecule for bacterial growth.
So they're essentially starving the bacteria.
In a way, yes. And isoniazid is another important antimetabolite. It inhibits the synthesis of mycolic acid, which is a component of the cell wall of mycobacteria.
And mycobacteria cause tuberculosis, right?
Exactly. So isoniazid is a key drug in treating TB.
Okay, so we've covered drugs that disrupt the wall, protein synthesis, nucleic acid synthesis, and metabolism.
Right.
Are there other ways antibiotics attack bacteria?
Yes, some antibiotics directly disrupt the bacterial cell membrane.
Which is basically the outer layer of the bacteria.
Exactly. Daptomycin and polymyxins are examples of these membrane disruptors.
Okay.
And then there's a newer class of drugs called lipid biosynthesis inhibitors.
What do those do?
They block the synthesis of fatty acids and lipids, which are essential components of bacterial membranes.
So another way to disrupt that protective barrier?
Exactly.
It's amazing how many different ways we've found to attack bacteria.
It is.
But of course, there's that whole issue of antibiotic resistance.
Right, that's a major challenge.
Why are bacteria becoming resistant to these drugs?
Well, bacteria are incredibly adaptable. They're constantly evolving.
Okay.
And when we use antibiotics, we're basically creating a selective pressure.
Meaning?
We're killing off the susceptible bacteria, but the resistant ones survive and multiply.
So it's like survival of the fittest.
Exactly. And these resistant bacteria can then pass on their resistance genes to other bacteria.
So it's spreading.
Right. And that's why antibiotic resistance is such a serious problem.
And how do bacteria actually become resistant? What are the mechanisms?
They have a few tricks up their sleeves. They can reduce their permeability to the drug.
So the drug can't get in.
Exactly. They can produce enzymes that inactivate the drug.
So they're basically breaking down the antibiotic.
Right. They can alter the target site that the drug normally binds to.
So the drug can't find its target.
Exactly. They can develop alternative biochemical pathways to bypass the drug's effects.
Clever.
And they can even pump the drug out of the cell using efflux pumps.
So they're basically throwing the drug out as fast as it comes in.
Pretty much. It's amazing how resourceful they are.
And it's scary. So what can we do about this resistance problem?
Well, it's a multi-pronged approach.
Yeah.
We need to use antibiotics more judiciously.
Meaning?
Only prescribing them when they're truly needed, choosing the right antibiotic for the specific infection,
Okay.
and making sure patients complete the full course of treatment.
So no skipping doses.
Exactly. And we also need to develop new antibiotics.
Because the old ones are becoming less effective.
Right. And we need to find ways to overcome those resistance mechanisms.
So it's a constant battle.
It is. But there are some promising avenues of research.
Like what?
Well, we're looking at new drug targets,
Okay.
new ways to deliver drugs,
Okay.
and even using combinations of drugs to make them more effective.
So it's a multifaceted approach.
Absolutely. And it's going to take a global effort to tackle this problem.
Absolutely. Now, what about antiviral drugs? Viruses are a whole different beast, right?
They are. They're much trickier to target because they rely on our own cells to replicate.
So it's hard to find drugs that kill the virus without harming our own cells.
Exactly. That selective toxicity is a major challenge.
So how do antiviral drugs work?
They typically target specific viral proteins or enzymes that are essential for viral replication.
Okay.
One important class of drugs is the nucleoside analogs, or NRTIs.
What do those do?
They interfere with the enzymes that viruses use to replicate their genetic material.
Okay.
AZT, acyclovir, and tenofovir are examples of NRTIs.
Okay.
Then there are the non-nucleoside reverse transcriptase inhibitors, or NNRTIs.
Which are?
They also target the viral enzyme reverse transcriptase, but they bind to it in a different way.
Okay.
Nevirapine is an example of an NRTI.
Gotcha.
And then we have protease inhibitors.
Okay.
They block viral proteases, which are enzymes that process viral proteins.
Okay.
Indinavir and sequinavir are examples.
Okay.
We also have fusion inhibitors, which prevent viruses from entering our cells.
So they're blocking the virus at the door.
Exactly.
Yeah.
Fuvertide is an example.
Okay.
And then there are neuraminidase inhibitors, which block the release of new virus particles from infected cells.
So they're preventing the virus from spreading.
Right. Tamiflu is a well-known neuraminidase inhibitor.
Okay.
And finally, we have interferons, which are naturally occurring proteins that boost the immune response against viruses.
So they're kind of like a natural antiviral defense.
Exactly.
Okay, so it's a whole different set of strategies for tackling viruses.
Right.
What about fungal infections? Are there many antifungal drugs? So they're disrupting the fungal cell membrane.
Exactly. Polyenes, alzols, and alaminas are all examples of ergosterol inhibitors.
Okay.
And then there are drugs that target the fungal cell wall.
Okay.
Echinocandins and polyoxins are examples.
So similar to the antibacterial drugs that target the bacterial cell wall.
Right. And then there are other antifungals with different mechanisms of action.
Okay.
Grisosulfan and 5-fluorocytosine are examples.
So a bit more limited options for treating fungal infections.
Yeah, but there are still some effective drugs out there.
Okay, and finally, what about parasites?
Antiparasitic drugs are a very diverse group with different mechanisms of action depending on the specific parasite.
Okay.
For malaria, we use quinine derivatives and artemisinin-based therapies.
Okay.
For other protozoan infections, metronidazole is often effective.
Okay.
And for helminth infections, we use drugs like praziquantel and mebendazole.
So a whole range of drugs depending on the parasite.
Exactly.
Well, we've certainly covered a lot of ground today, from those immune system malfunctions to the incredible ways we're fighting back against infections.
Yeah, it's been a really comprehensive deep dive into the world of immunology and antimicrobial therapy.
It's amazing how much we